Hopefully I can live up to that introduction. Thank you so much, Alex. Okay, and as some of you know, I changed my talk because of what Stephanie said yesterday, which was like, I heard a bunch of talks and I'm like, oh shoot, I want to do something different for this workshop. And so I changed my title, which is basically how I came through my work with Vincenzo over the past 5 years, really to think about how do we take our imaging data to completely change the way we think about how to build models of cellular machines. This whole spirit of this conference. Okay, so I'm going to start sort of pedantically, right? Imaging data has been really important for building descriptions of matter forever, since we've developed ways of characterizing shape and dynamics at all different scales. This is a beautiful image movie from my colleague Sid Nagel, which is just a splashing water drop, right? So you can capture the dynamics of this drop, the dynamics of drop breakoff and splashing, really to understand the origins of fluid dynamics, right? So if we think about how we understand condensed matter, right, there's a bunch of molecules that come together in this condensed phase that have certain properties that then describe how these this blob of condensed matter responds in response to external forces to create motion, right? We're here to study biological living matter, right? So as soon as we add more complexity of biomolecules and internal energy, right, there's new properties that emerge from these types of condensed matters. But more importantly, there's all these feedbacks that make our understanding of how this matter moves and changes in response to mechanics, our understanding of it as a machine or piece of matter, really complicated, right? And I think the thing that's I've been struggling is with how do we actually learn these feedbacks in a quantitative way to actually make forward prediction of the dynamics of motion. Okay, so so I'm going to focus, or my sort of obsession recently is sort of on the feedbacks, right? Like if we think about internal stresses, the field of active matter has sort of been developed for at least a few decades now. We sort of know how to think about active stresses, but it's really how does a property with a certain that can coarse-grained with an elastic or viscoelastic parameters, how those viscoelastic parameters change over time. Okay, so this is sort of what I think of as the challenges. We've, over the past 2 decades, we've gotten really great ways of capturing dynamics across scales of different processes from the subcellular to multicellular. So we can capture dynamics. But in a conversation with my friend Cliff Brangwyn, I made a comment that our understanding of cell mechanics is way closer to geophysics than it is to modern condensed matter. And what I mean by that is like, we don't really have that much ability to control it. We can engineer some genes or some molecular activities at one scale. We don't know how to control its emergent properties in the same way of synthesizing new type of behaviors or reconstituting it in a way that we can't do that for geological formations, right? So we would like to be able to do this to engineer morphogenesis or engineer cell dynamics across scales, right? To be able to control this. The good thing is, over the past 50 years, we have a molecular basis of living matter. No need to introduce this room. And we know a lot about the molecular scale biophysics, but we need to understand how these all work at the system scale. Okay. All right. So the big challenge, I'm assuming that keeps most of us up at night, is really how do we bridge these scales from collections of molecules all the way up to collections of different cells interacting to build tissues. This is a really complicated problem because all these systems have different feedbacks at the different scale, but also bridging scales of space and time, right? So I'm gonna go back down a little bit and think like the way that I've been thinking about this, the way I'm going to talk about in this talk is really think about the systems that are controlling adherent cell migration at a single cell scale. So going in the subcellular scale of what Chase spoke about, where we really need to think about how does actomyosin self-organize and build contractile machines across the cell that control the tension that is regulated at the cellular scale. And how do these cells then build adhesions to the extracellular matrix to enable motion, right? So both of these subcellular assemblies or organelles that have force responsiveness change in their molecular composition over time. As they change their molecular composition, they change their mechanical response, right? And so, I spent 20 years studying how these processes change, these feedbacks between mechanics and biochemistry, right? And of course, we can study these processes in live cell images, either by expressing fluorescent constructs of proteins. But the fantasy, I'm assuming most of us in this room, is like, how do we actually get understanding from these kind of movies, right? So if I can take a tag of a fluorescent protein, how do I make a prediction about how the mechanobiology of how that organelle is functioning at scale, and how does it actually control adhesion and migration at the level of whole cells? In the case, again, going back down to the level of the problems that we've been studying on, we care about what regulates the magnitude and spatial distribution of cell adhesion and tension. How does the actin-myosin cytoskeleton maintain its structure despite varying degrees of tension in response to internal stresses, external stimuli, and execute shape changes smoothly? Right. I often say that one of the big differences between actin-myosin you put in a cell that's in a cell versus what we can reconstitute in vitro is that it never rips itself apart unless you really do something bad to it. So it's always able to execute these like physical changes very smoothly. And this is necessary for cell physiology. Okay. Okay, so how do we normally study this, or how we still do study it this way sometimes? This is an example from a collaboration we had with Alex McGillner. One approach we all are familiar with is like a microscopic agent-based approach, where we throw in all the collections of what we know about the microscopics of every single molecule. We try to do our best in careful experiment to make measurements. As I think most of us are familiar with, these break down very quickly, right? They have very limited prediction in time. There's always too many agents. Some people still think that we could measure every single molecule activity and we'll understand how the whole systems. I don't believe that. It's really limited in how we're going to be able to make predictions. On the other extreme is like what the physicists field is like the best way of doing things, which is like a continuum physics-based theory, which is great. But they're built oftentimes on foundational models of non-living matter, right? So you're missing new types of interactions or potentials. They completely lack any biochemical reality or function, right? And so there's really challenges with understanding these adaptation rules, the spatial temporal variation that occurs that we know is important for cell function, right? All right, so both of these are problems, but they illustrate one of the challenges with our field, which is really, it's a coarse graining problem, right? It's like, how do we actually properly reduce the dimensionality from this system, from this massive collection of molecules, down to something that is predictive? And Chase brought this up really beautifully with his studies of cell migration. Like, how do you take, and I'm going to say, like, how do we take other kinds of imaging data and other ways to do this dimensionality reduction? Okay, so as an example, so the way I guess we've gone about this is, I'll just be honest with you because you're like a small group. Like starting in 2017, Vincenzo's like, do you have data? And I said, sure, I can give you data. And then he would say, well, I'm going to build a model that predicts something. I'm like, I don't believe that's going to work, right? And then so we just kept working with problems that like I had studied enough that I trusted when it was bullshit, right? So we did this with several different instances. And so when we got to this problem of cell mechanics, like it was a problem that we had studied for years, right? We had studied how to adherence, like kind of a boring question is like, you have an adherent cell spread on a surface. You measure the forces that cells impinges, like how is the spatial and temporal variation of those forces regulated? Okay, so we had made tons of measurements, had tons of data. And sort of the best model we had been happy with was one we had built with Christina Marchetti and Sheila Banerjee back in 2015, which is just a continuum active solid model where you just treat the cell as an active elastic gel. And you treat it with uniform adhesion, and just because of the geometry of the cell, it will impinge forces at the boundary. Okay, so this did a very good job of making predictions, both in controlled geometries, but also in the wild, in some cases, of what the distribution of traction forces were at the whole cell scale, and gave, with two interpretable parameters of an elasticity and an active stress, some understanding of what regulated forces transmitted by cells. Okay, but obviously this is really bad at predicting anything that's changing in space and time. It doesn't give any information of what's happening at the site of a single focal adhesion where the forces are actually getting transmitted. It's not giving an understanding of how forces are changing over time or how the localization of these molecules of paxillin or actomyosin might be important for regulating forces. Okay. All right, so there's lots of things missing from this continuum model. So in this, we basically took all the data sets that Patrick had generated as a postdoc in my group, but then also that we could generate at large scales in our labs on a lot of different cell types. So that was all done by Stefano and then Shailaja and John Devaney in my group. And then two brilliant students, Matthew Schmidt and Jonathan Colin in Vincenzo's group, worked together to ask a question as like, could we train a neural network to make a prediction to go from the images of proteins to a traction force field? Okay, so this is the same kind of modeling question as like, how does a distribution of proteins make a prediction of the mechanical output of the cell? So he used a convolutional neural network architecture, which not surprising, and with the capabilities of like thousands of images, basically taken from time-lapse movies of paired images of images of paxillin and traction forces. They were able to successfully train a convolutional neural network to make a prediction from the Zixin images to force field. And the loss was able to be minimized. Okay, so you can go through and ask, does the error in how well you can predict the traction force depend on whether you're in the training set or your data or your test set? And this is just showing that the mean squared error really doesn't matter. There's some variation here, but you're able to predict forces in your test set as well as you could if your cell was in the training set. Probably one of the more surprising results for me at the time was our training set consisted of all wild type cells with high amounts of contractility and fibroblasts. And the question is, if you do a really strong perturbation to the cell and change the amount of force output, would it still do a good job at prediction? And the answer is yes. So this is a movie of a cell that you've treated with saturating amounts of a Rho kinase inhibitor. So at the point when you wash in this drug, the traction forces decrease. The distribution of Zixin intensity also changes dramatically, but still your model is doing a very good job at predicting the changes in traction forces across the cell. And I guess what that started to indicate was that, for a lot of time, the way we had approached trying to build biophysical models of this cellular machine was to sort of engineer the environment, make the cell more reproducible, put them in the right geometry, make the experiments more and more reproducible. And this was sort of the first evidence that the variation in our biological data sets was actually a feature and not a bug. Is that when we had our cells just randomly trained, that there was so much variation in the adhesion intensity or distribution across the training set that it was actually building a really robust model for prediction, that it was robust against strong perturbation. And I would say it's not something that we've explored quantitatively, but Vincenzo and including Matthew and Jonathan went on to do another project on a developmental dynamics of gastrulation and fly, right? And there it's very canalized and robust, and the model was much more sensitive to variation, right? So I always, people always sort of look down on me for using tissue culture cells, and now I feel like I can think that the variation in tissue culture cells is actually a feature, not a bug in this case when in the machine learning era or model approach. Okay. Okay, so once you have this trained model, you can think of it as a simulator of predicting from one type of field, in this case, a protein distribution field of Zixin, to a field of biophysical measurement. And so we can ask what exactly is in this image of Zixin that makes it a good predictor of the force field. So you can ask what would happen if you trained a model with just the cell mask, or an image of all the mitochondria, right? And you can start to see that the error and how much your error is between what you of your trained model is it increases a little bit with just the cell boundary, which again is not surprising given that we built an entire model on cell geometry. So we knew cell geometry was a very good predictor of forces, but that when you image for mitochondria, it's not a good predictor of the force field, which is also not surprising. And we could do this for different types of adhesion proteins, right? And also actin-myosin, and ask, what is the change in the intensity? Now, or what's the change in the error? And I will say that I was talking to somebody recently who has done a lot of work on digital staining, which is this general idea that you can image something in one channel, say one protein distribution, and predict another signal, right? Say a phosphorylation state, or you go from phase contrast images and make a prediction of where your mitochondria are, where your nucleus is. And it seems to me it seems to be it seems to be true so far that in most cases that if your training set is sufficient enough, you're always going to find one convolution from image A to image B, no matter what those two images are. And I think this is an interesting question for those of us who like imaging, of like, how much data do we actually need to take in terms of pairwise images if we can actually get a nice transformation from one imaging field to another imaging field. So I think there's a lot of room for us to explore this further. Yeah.
I don't understand what the error is an angle means.
This is the best measure of the error was the angle between the forces, because it was a measure of that Matthew used for error of the force direction.
Okay.
Yeah.
Does it depend sensitively on whether you coarse grain? Like, what do you count as like a point where you measure the force?
Oh, these are all images, right? So they're all like sort of pixel-based fields.
Okay.
Yeah. So the traction force measurements.
There's one pixel where it's 50 degrees to the left, and then the pixel next to it is 50 degrees to the right.
Yeah. So it's learning a vector field, actually. Each pixel has a vector associated with it. And it is true that the magnitude then it impacts the directionality. The directionality is much more sensitive. Yeah. Okay. Okay. So now, so then one way you can use this simulator, if we think of this trained neural network as a simulator, to say, take an input of an image where now we can change, say, the intensity of our pixels, the distribution of something that looks like a synthetic focal adhesion, or the density, and ask, what's the prediction of the force field? So this is just a simple-minded simulator from our trained neural network. And then because it's simulated data, we can do this much more rapidly than what we could do experimentally, to ask what happens if we change morphological features like we would expect to be with adhesion sites in a cell. So this includes like the aspect ratio of this feature or the total area. And what was, in this case, we're keeping the density the same because that was too complicated. But what you can see here is that there is a peak in the total amount of force output around that black box right there, which tells you that the maximal force output at an adhesion has to do with a certain aspect ratio and area, which I'll tell you, after studying how force is dependent on adhesion area and intensity and shape, like this area is the same location, the same distribution of morphology that we would have seen from experimentally, that we spent 4 graduate students trying to make these measurements, that you can extract out this correlation nicely using this simulator. So if this were something new that we hadn't studied before, this could be used as a way to hone in on where to look at sensitivities in my experimental measurements in a way that I wouldn't have known previously just looking at random correlations. The other nice thing is now this simulator can be used to quantitatively look at the force sensitivities, to look at how the force at the adhesion site depends on the intensity of the protein intensity locally for a varying focal adhesion length. And so what you can see is that when the focal adhesions, synthetic focal adhesions are small, there is minimal force sensitivity, but it's not until the adhesions get longer that you see this increased force sensitivity. And again, this is something similar that we had seen for force-dependent changes in Zixin accumulation experimentally that comes out in this simulation data, right? So this is a tool for experimentalists to use as a way to refine our hypotheses about how the mechanobiology is working, about how correlations between, say, protein concentration and forces are changing as a function of a morphological or shape process. Okay. All right, so taking a step back, right, I, as an experimentalist, am sitting over here with generating data. I guess I always mostly trust the data we take as a process that's happening in the real world. Then we can take this data and build a neural network model to make a prediction of one data field from another data field. And that's another type of model or simulator that we're using to make accurate predictions, but it still is not interpretable, right? I think there's lots of utility for using these kinds of predictors, but it's not interpretable in the way we'd like to biophysically, right? On the other extreme, I'm just showing you what the predictions from our continuum model were. This is this coarse grain model I showed you in the beginning, where it just showed that there was very limited localization of traction forces across the cell. So this is where we are with physics continuum theory. There's this big valley of gap of understanding between the two. So how do we bridge this? Okay, so I'm just going to walk through two things that we did to make the neural networks have more physically interpretable, but then also augment the physics-based theories with some biochemical insight that we can then test with our neural network. Okay. All right, so in the case where you have a process, say you show that you have process field A and field B, and you know field B is can be predicted from features in field A, but you have no idea what the physics is. So this is completely agnostic. It's like trying to understand the basis of understanding what kinds of equations would be sufficient, sort of in the static version of what Chase was doing with dynamics, but now it's just distributions of intensities, right? So in this case, what we're doing is we're going from an intensity of just protein distributions. We're trying to predict a biophysical field. And the question is, what's the basis set of expressions? And here are Green's functions that tell us the mathematical constraints on what is needed to go from this data field to the biophysical output, right? And so here, what we do is, as we're training the neural network, we can put in a Green's function layer that learns both the nature of the Green's function. So in this case, we found that we could semi-accurately repredict the traction forces with two different Green's functions, the yellow and pink. The yellow one is a little bit long range, sort of smells like elasticity. The pink one is very localized, sort of smells like a localized adhesion, right? Localized adhesion, right? And so then we can learn where the sources are of those two Green's functions and their contribution to the overall traction forces, right? So this force prediction, this Green's function neural network, is only using math to go from the data field to the predicted vector field, right? There's that. So it is fully interpretable if you like math. All right. There's no physics there, but it gives you a set of mathematical expressions to use. Okay.
Yeah. But is this a little bit similar to what Chase was doing in that, like, you make some expansion and basis functions, and you find it's sort of,
I like the way you showed it with also all the error, but it was sort of the same thing as like, how much does your error go up if you minimize the number of expressions you use? Yeah. Okay. Okay, so on the other approach is that you could take a physics model that you have and extend this sort of continuum theory by making some constraints placing constraints on your neural network training. So in this case, if we think about training our neural network to go from protein distributions to forces and minimizing the loss, what we're doing here is putting in a physical bottleneck here, where we are putting in a PDE solver and constraining, saying that you need to obey this law of physics and learning the parameters that you need, depending on which ones you're letting are tunable, and learning and basically doing parameter estimation on that model. Okay. And so here, what we did was we So all the update rules obey this rule of physics with the caveat that you can have a continuous variation of your variable, which is a big caveat for data fitting. I'll just say, like, it is a human judgment about what parameters you choose to let vary and in what way. Okay, so in this case, we took our trusted continuum theory of the cell being a stressed elastic sheet with continuous adhesion, right? So that's our physics. And then we allowed certain parameter variation. This took a lot of iteration between all of us to decide which parameters we decided were okay to vary. In this case, we only allowed adhesion to vary and allowed the model parameters of active stress and elasticity to depend on the Zixin intensity. That's the field that you're measuring, okay? And so now you're just learning the parameters from the imaging theory. You're not changing the physical model. So in this case, we can learn the spatial distribution of the adhesion field, and then we can learn how the force depends on the Zixin intensity, which again, I haven't gone into the biology of Zixin, but Zixin is a force-sensitive protein, and so its intensity is dependent on the local stresses. So this all made sense biologically. Okay, so this is a way to estimate parameters in a spatially varying way when we're just thinking about statics, of going from a distribution of features of an intensity distribution to making a prediction about something that you'd like to understand. So it augments our continuum theory with spatially and in theory time-dependent variation of biochemistry from the imaging data itself that you get from the images, which has also been a struggle for connecting experimental measurements to theory. Okay, so these are two different methods that sort of start to fill in a gap, these physics-inspired neural networks and physics-constrained neural networks between this idea of just these agnostic deep neural networks and sort of our traditional ways of building theoretical models that I think are really useful in terms of experimental constraints on building models. Okay, and I think what I'm super excited about is that, you know, this one small project, right, really started to change the way I think about how to build models, right? How to build data-driven theories, and I guess everybody in the room is in the same place because we're here together. So I think it's super exciting. So this is something we've been trying to start up at Chicago, go through the NITMB and our Physics Frontier Center, right? So we've been starting to think about how we can broadly use imaging data to learn cell dynamics. This is something done with collaborators Aaron Dinner, Suri Van Kuntenathan, Vincenzo Vitelli, and staff scientist Leah Ding, who actually came trained from here, both in the Allen Institute and then Gauden's lab. Okay, so I'm going to just give 1 to 2 slides of this, of really where these are projects that are ongoing and how we're thinking about this. First is really going to dynamics, right? All I showed you was statics, going between 2 different fields. So how do we learn interactions and forces from movies? How do we actually use that to then learn mechanochemical feedbacks and adaptation rules that occur over time or that are force responsive? Something I won't talk about, but this general idea of digital staining, which is interesting for me as an experimentalist because it affects how much data I need to collect and what type of data and what I can learn from that data. And then once we understand these dynamics, both in a static and dynamics at a subcellular scale, is like, how do we build entire representations at the cell scale, right? So this holistic from the Allen Institute state representation from these imaging data. Okay, so Billy, who's here, is a postdoc with Aaron and Suri, and so she's been looking at data collected in my group from Amanda Johnson and soon to be Victoria, who's here. This is movies of cell migration and a model system of gastrulation. So it's also cell dynamics movies like Chase. So Billy's really interested in how do you build collective models of cell-cell interactions. So if you want to learn more about this, you should go to her poster. I want to show one experimental data because it's sort of like the extension. Both these are sort of like extension of what Chase had showed. Where you have movies. These are mesodermal cells of stem cells that are differentiated, sort of right post EMT. And what Amanda has found during her PhD is a GPCR signaling that affects directed cell migration of these mesodermal cells that helps them spread during gastrulation, right? So we want to understand the interaction potential of these cells during gastrulation, but also what their long-range dynamical cue is that lets, I didn't show it, that lets the cells in the upper left, all the cells migrate towards the center of this model system. Another question is, these are dynamic. So now these are all movies of Zixin, right? So I showed you our story of static, of predicting Zixin images to force fields. Now could I predict in time how cytoskeletal features and organelles are going to change and adapt in response to time? So Zixin is part of a force-sensitive feedback. I'm going to skip this, where this is a model of a stress fiber. So if the stress fiber, the actomyosin stress fiber, receives variation of external tension, Zixin is essential for repairing the stress fiber and to reassemble actin at the site of damage. So it's a dynamic process that allows for stress fiber repair within 30 seconds. So this is a time-dependent process that is responsive to sort of local strains within the stress fiber. And so again, Patrick sort of painstakingly during his postdoc had studied the response of the sort of mechanical response of stress fibers using optogenetics, where he applied local stimulus and measured a response of deformation of the stress fibers. And he found that this Zixin-mediated repair response in collaboration with Ulrich Schwartz's group was sort of essential for the underlying elasticity of actomyosin stress fibers. So if we go through in conventional ways of looking at the deformation fields of the stress fibers and modeling these as an active viscoelastic medium and asking then what the viscoelasticity is, this stress fiber behaved like an active elastic solid, okay? And without this Zixin-mediated stress fiber repair, basically the whole system changed. The tension in the system did not get impacted, but the whole system became like a fluid. So this is now a Zixin knockout cell where you still see stress fiber. There's no stress fiber repair. There's still a lot of contractility, so the contractility is there. But now the whole system has a very rapid relaxation time. Okay, so this took us like 5 years to do these experiments. It took us like multiple groups to do this model. So our fantasy is, well, we have all these movies. Would we have been able to learn all this stuff without going through the painstaking process of doing optogenetics and just looking at the internal fluctuations of the Zixin in the time-dependent movies? So, let me just give you something else before that. That's one fantasy. And then just as an example, like this feedback, right? This is something like, this is the level of this qualitative diagram is the level at which we understand these feedbacks. Like we want to turn these into more physically correct and quantitative models of feedbacks to understand really what the time scales is, what the coupling coefficient is, by which stress fiber repair is controlling stress fiber mechanics, right? And that's something that, again, in terms of turning these materials framework into a dynamical systems framework of adaptation is something that's necessary. So Kwang Hoon, who's also here, is his dream, who's his student with Aaron, is to now take these movies, these time-lapse dependent movies of Zixin changing over time to try to learn what these dynamical laws are of actomyosin and what is predicting their forward time evolution, right? And so we can use the models that we had developed with Ulrich, right? These continual models of active viscoelastic fluids, but now use the parameter estimation approach that we had in the static system to actually learn what the forward time dynamics. So this is something that's just ongoing. You can talk to Kwang Hoon at his poster as well. And sort of like, if I just think about this fantasy, right? Like, we want to take all these machines, like whatever machine I care about, and like I'm used to like dissecting it molecule at a time. I'm also used to, as an experimentalist, like finding something that I want to measure and then saying like, okay, is that the important thing for my theory collaborator to measure? And no, it's not. Let's go try something else, right? It would be nice to have procedures to be able to know which length scales to focus on, which molecules to focus on, right? And so building this pipeline in which we can take all this imaging data to more rapidly advance theory discovery would be very exciting, right? And so, many of us in this room have spent a lot of time trying to develop active matter theories. But I think the exciting thing for me is really thinking about how active matter is adapting over time, right? What are the time-dependent changes? Why, in response to some stimuli, there's like a homeostasis response that allows stress fibers to repair versus assemble and disassemble. Like, what is the difference between those cues? So that's really requires developing sort of a framework to understand adaptive matter. And including all these mechanochemical feedbacks that exist. I think I'm just going to skip over this because I'm running out of time. Am I running out of time? What time did I start?
You're equivalent 3 minutes.
I have 3 minutes. Okay. Yeah, so that's exciting. And then the last thing I'll just say is that, with Leah and Annabelle is like, so you take all these images. I think for me, if I think about biophysical mechanism, I would like to learn something at the scale of the cytoskeletal mechanobiology. Like, I won't be satisfied to learn it as friction rules. Like I'd like to go down to a subcellular scale to understand the rate of endocytosis is changing, and it's changing in a force-dependent way, right? So what we're thinking of is like building up this mechanical picture in the cytoskeleton, but then getting a representation at the cell scale that tells you sort of a mechanical state of all these organelles. And focal adhesions are like a great example of a mechanobiology organelle where, this is like from a review article 5 years ago, right? Where you have all these different features, like they change their morphology, they change their function, they change their composition, they change their different mechanical state, right? And so all this understanding of focal adhesion dynamics and mechanobiology was built up from 20 different labs. And yet we don't understand the coupling between the mechanics and the signaling and the composition in any kind of quantitative way that prevents us from saying, how does this mechanobiology actually impact cell function, right? To make sort of cell mechanics more quantitative in that way. So our idea is like, could we do the same thing that we did for the force inference, but then now predict using this idea of digital staining from Paxillin images to, say, a signaling state or a holistic composition of the focal adhesion? Like, how can we build up sort of these models of understanding this multimodal sort of understanding of different composition, concentration, and mechanical sort of characterization of the focal adhesion? So Leah's been doing this just from Paxillin, asking the question just from images of Paxillin is like, if I have an image of Paxillin, could I represent it as a lower dimensional variational autoencoder? Like, can I reconstruct what the images are? And the answer is it does a pretty good job, right? So you could take this image of Paxillin, this focal adhesion, and then say, like, I can represent the morphology and the intensity by this 8-dimensional variational encoder, and then look at this in this latent space, right, of the first two, dimensions, right, and ask, what features am I picking up, right? So here on the lower left-hand kind, you have things that sort of smell like nascent adhesions, right? On the lower right-hand corner, you have things that look like larger, mature focal adhesions. So is it possible for us to map in this higher dimensional space changes in composition, concentration, as focal adhesions but undergoing its maturation, right? And then you can imagine once you have that characterization, can I build up a representation at the cell scale of each voxel or pixel of different mechanobiological state of the actin and the focal adhesions to build up the mechanical, like a representation at the whole cell scale, right? And so I think this gets to like a longer term fantasy. It's like, we want to understand how cells or tissues process information, right? And to understand that, you need to understand how all these different little organelles are doing that, right? So would this give us a way to tackle cellular mechanical information processing in a way of sort of characterizing it from our images, but also making predictions about what cells are going to do over time, akin to sort of like, a fantasy of a Waddington-like landscape for differentiation, but one that captures morphodynamics. Okay, great. Thank you. Yes, and I'm very grateful to work with wonderful, wonderful collaborators and students without which this work would not be possible. So thank you.
Hi, really nice talk. I was just curious about the model that way you predicted the traction map from the protein images. I was wondering if you had to also predict the substrate stiffness that it is on.
Yeah.
That you need to input any other thing into the model, or
Yeah, we spent a lot of time. I was really like, does it matter who takes the images, on what microscope it takes the images, and how much environmental changes, if it doesn't matter on the cell type. It turned out to the ability, the caveat that we were predicting relative tractions that go between sort of like a rho kinase dependent inhibition to like wild type. These kinds of variations did not force us to retrain a model, that we could use a single trained model to use a variety of cell types. Now, is there going to be some perturbation that breaks it? Absolutely. But we hadn't found it yet. We also haven't, we haven't hit it hard enough to break it.
I was just thinking between your work and Chase's work, if there is a way to kind of use the images to come up with some state variable, dynamic state variable that you can kind of then use in that framework to combine the world of imaging into this particle world. Yeah,
that's something that we've definitely been trying to think about. I think people have controversial feelings about this. We should have like a beer over this. I mean, I do think it's really fascinating. Like one thing that we've talked a lot about recently in terms of model building with dynamics is like, is our images are being like a lot of times, like the convolutional neural networks are very nice because we don't have to do any pre-processing. And that pre-processing or segmentation or analysis always adds noise to the system, right? It's adding some kind of noise in terms of measurement. And so if we could do it in a way that did not require that, sort of akin to the convolutional neural networks, it would be really nice. Like, I've been feeling that way definitely for statics. Like the segmentation just really, anything that's not like membrane bound, I'm like really strongly feel that segmentation just adds more complexity than it needs to. And I think that's really exciting. So one thing that Susan I know really doesn't like, the old school cellular POTS models is like one thing that's come up, which was like this. What?
I don't not like
it. But it's like this one idea of like this Ising model that's on a pixel grid that you could imagine asking, like in your pixels of your image, like, what are the interaction potentials of these pixels? Yeah. Yeah, I don't know. The question of when you need to segment, when you need to know it's a cell, when you need to know it's a tissue. Like, I think it's fascinating. I totally think it's fascinating.
You don't segment, but don't just do voxels. So yes, anybody who wants to have a beer, we can all.
Yeah. Well, and like the whole digital pathology field, right? They don't care that cells exist. Right? They just get voxels of histology sections, right? And they're making predictions about distributions at the tissue scale. I find it totally like, at what scale do you actually need to worry about entities existing versus it's just some field?
Right.
Yeah.
Right.
So the physics constraint model didn't work as well as the neural network, right? Did I get that?
Yeah, that's correct. Yeah.
So did you think about what physics is missing in the physics constraint model? Like, are there things you can add to how it competes?
I'm sure there are. I'm sure there are. We were very nervous about overfitting the parameters. Does that make sense? Like, and I think if somebody wanted to work on this further, I would want to know more about which protein distributions were useful for making that prediction versus not, like that variation of like Paxillin versus Zixin. Like I have reason to think that Zixin should do better, but is that the case? There's definitely more. The parameter fitting there makes me very nervous. So I think for people that are very smart, figuring out how to do this in a robust way that is consistent between groups, I think is really important. Yeah.